Hello everyone, Assalamu alaikum. Welcome to the new lecture on medicinal chemistry. I am Faraz Qurban Rajpur, and in this lecture I am going to discuss about the structure based drug design. That is one of the famous method of the computer aided drug design. These are the outline of this lecture. At the end of this lecture, you will be able to learn about computer aided drug design, different types or approaches of computer-aided drug design, virtual screening process, structure-based drug design, and the different approaches or methods that are used in structure-based drug design like de novo drug design, molecular docking. At the end of this lecture, you may also find a quiz for your self-assessment. Computer-aided drug design. Computer-aided drug design involves all the computer-assisted techniques or application of different computer software to discover, design, and optimize biologically active compounds with the evidence used as a drug, having the desired structure and properties. So computer-aided drug design, we have already discussed in detail that these are the different applications of computer software in which we are going to discover, design, and optimize different chemical compounds in order to make them effective in the treatment of different diseases. Major type of approaches in the computer-aided drug design. Basically, there are the two main types of computer-aided drug design, or there are the two main type of approaches for the computer-aided drug design. One is the structure-based drug design. Structure-based drug design is also known as the direct approach or direct method. Second is the ligand-based drug design. Ligand-based drug design is also known as the indirect approach or indirect computer-aided drug design method. Virtual screening. Virtual screening, as the name indicates, is basically related to the screening of something that is done by the help of different computer-aided program or computer-aided software. In case of drug discovery and development process or computer-aided drug design, virtual screening generally refers to the screening of different chemical compounds in order to evaluate their properties or in order to evaluate their biologically activities to check that whether they are effective in the treatment of certain diseases or not. Basically, the screening is done on the basis of their chemical structure, different functional groups that are present in their chemical structure, and their compatibility with the particular drug target like receptor, protein, enzyme, and other drug target is checked. So in virtual screening, basically medicinal chemists are going to discover about the hits and lead molecule. In this virtual screening process, a large database of compounds is available in the software, and from those different variety of chemical compounds, only few compounds are selected as hit. Hit are the compound which shows the particular affinity toward the selected drug target or the disease which we are targeting to discover the drug molecule. Virtual screening is becoming famous nowadays as most of the pharmaceutical companies are using this program for the discovery of the new lead molecule for the designing and discovery of the medicine. There are mainly two types of the virtual screening. One is the structure-based virtual screening. Second is the ligand-based virtual screening. Structure-based virtual screening is based on the structure of the drug target, that is the protein or receptor, and the structure of the active sites, means the amino acid sequence and the available active site for the drug target. Whereas on the other hand, ligand-based virtual screening is based on the techniques or the different method of the ligand-based drug design, and it mainly rely on the knowledge of ligand, and it uses different ligand-based virtual screening technique like pharmacophore modeling and the QSAR approach, in which in pharmacophore modeling we are going to check about the similarity between the drug molecule from the database. Now let's discuss about the structure-based drug design. Structure-based drug design is one of the method of computer-aided drug design that is based on the three-dimensional structure of the biological target. We know that there are the different biological target for the different drugs. Like protein is one of the famous biological target for the drug molecule. Among the protein drug target, we have the receptor, enzyme, and many others. So basically, when scientists are using the structure-based drug design approach for the discovery of new drug molecule, the most important thing is to get knowledge about or to get know-how about the entire three-dimensional structure of that particular biological target like receptor, enzyme. These structure can be evaluated through the different techniques like X-ray crystallography or NMR spectroscopy, that is the nuclear magnetic resonance spectroscopy. There are the mainly two main methods that are used in the structure-based drug design. One is the molecular docking, second is the de novo ligand-based drug design. The second method is mainly used for the discovery or the design of antagonist, agonist, and the inhibitor type of the drug design. Protein structure determination. The most common method that are used for the identification or determination of the three-dimensional structure of protein are the X-ray crystallography and the NMR spectroscopy. These both are the experimental methods. Okay, so there are the certain conditions in which these methods are not successful due to certain challenges. For suppose, as in case of X-ray crystallography, to determine the structure, the protein must have the property to get crystallized. But there are certain proteins like membrane protein which are very difficult to crystallize. So in such cases, in case of membrane protein, this X-ray crystallography is not successful. And we know that majority of drugs, like 60% of the approved drug targets or proteins, are based on the membrane protein target. So in these conditions, like due to these experimental challenges, when these methods are not successful, then ligand-based drug design technique can be used as an alternative for the discovery and design of new drug molecule. Overview of the process that is involved in the structure-based drug design. So basically this is general overview or the different steps that are involved in the structure based drug design. First of all, the identification and the validation of the target means the identification of the protein structure. In that we have used the X-ray crystallography and the NMR spectroscopy. Next step is the analyzing of the structure for the potential ligand binding site, then lead identification, molecular docking, lead validation and optimization, and then the clinical trial. So first of all, we have to identify or determine the protein or the drug target structure. In the next step, we will check the structure, okay, and observe or evaluate the different binding site on which our ligand can be bind. In the next step, we will identify the potential lead molecule, and then we will perform the structure based drug design method like molecular docking, lead validation and optimization, in which we may modify the chemical structure or other certain changes can be made in the lead molecule to make the structure based drug designing more effective. And then we will do the different studies to check the effect or the biological activity of our new drug molecule on the living organisms, just like we will do the clinical trials. Structure based drug design is basically not a single step process. It involves different multiple cycles or different steps in order to give one successful lead molecule that can be used in the clinical trial. So as in first cycle or the first step, we have discussed that it involves the identification of protein. This involves basically the sub steps: isolation, purification of those protein, crystallization like in case of X-ray crystallography, and then the determination of the different binding sites. Okay, so these are the this can be done by the different techniques like X-ray crystallography, homology modeling, or NMR spectroscopy. In the next cycle, these compounds like different hit molecule which we have selected through the virtual screening process from the different database sources, and these selected hit molecule are now evaluated for their binding affinity with the target binding sites. And now those compounds which show the high affinity or affinity toward the binding sites are scored or ranked like in their interaction with the target protein or the active site is checked. That is basically dependent on their chemical structure and the different functional group of their chemical structure, because there are the different physicochemical properties which can be affected by such functional group, and due to those properties their binding with the target can be increased or decreased. Like there are the different groups which can enhance or decrease the hydrophobic lipophilic property, and there are certain functional groups due to presence of which certain ionic interaction, electrostatic interaction can occur, and the steric hindrance can occur in case of presence of multiple bulky group in the chemical structure. Now in the next stage of structure based drug design, medicinal chemists have to observe or determine the structure of the complex. The complex is usually the lead molecule that is effective on the binding site. And now this binding complex of lead or drug molecule or selected chemical compound with the receptor or the protein target is now being observed. Okay, and now on the basis of this complex structure, their biological activity of this potential lead molecule can be evaluated because it has already shown the affinity towards the binding sites. Okay, now next we have to identify that whether this effective binding has been successful in order to show the biological activity or not. So we will perform the different biological assay in order to perform the biological activities, like we will perform the different in vitro studies in which we have to identify the biological activity, like by observing the effect at the minimum concentration of this lead molecule, and we will observe the potency of the new drug candidate molecule on the basis of its concentration. And once the particular lead molecule has successfully shown the optimum biological activity, next will be the synthesis of the same type of other lead molecule, and their structure can be optimized in a particular way, which in order to show the maximum biological activity, to show the optimum interaction with the protein or the binding sites. Structure-based drug design and the HIV drug discovery. Structure-based drug design has been successful in the discovery and designing of the variety of different new drug molecules which have been approved by the FDA, and now they are available in the market. Like, structure-based drug design has been successful in the discovery of drugs that are targeting the AIDS disease. So these are basically the HIV drugs, and the prime target for these drugs is the protease. In the earlier 1990s, many approved HIV protease inhibitor has been developed for the treatment of the different HIV infections. So these protease inhibitors are basically developed or designed, discovered by the help of different technique of structure-based drug design. Like in case of protease inhibitor, mainly molecular docking method is used, and we know that molecular docking is the method of structure-based drug design, computer-aided drug design techniques. So among these approved HIV protease inhibitor, Secunavir is the first HIV-1 protease targeted drug that is available in the market. So this is the diagram for the structure of the Secunavir, that is the protease inhibitor for the targeting the AIDS or HIV infections. The first part of this diagram shows the structure of complex of the drug molecule and the HIV-1 protease. Okay, and the next part of this diagram is showing the chemical structure of the Secunavir, that is the HIV protease inhibitor drug. Now let's discuss about the different methods of structure-based drug design. One of those methods is the de novo drug design. De novo drug design approach of structure-based drug design is basically based on the structure of binding site. Means it is not relying on the overall structure of the receptor or protein molecule, but it only needs the structure of the binding site where different amino acids are present. So, if medicinal chemist or organic chemist who is interested in the discovery of new drug design, and if he knows the amino acid sequence of particular protein target, then he may design a new drug molecule on the basis of that amino acid sequence, that how these amino acids are positioned and which type of interaction or bonding can be involved with these amino acids for the different chemical compounds. So on this basis, he may design a new drug molecule and evaluate their different functional group which may be effective in interaction with the amino acid sequence or amino acid that are available in the binding site of the target. De novo drug design is successful in the discovery of thymidylate kinase inhibitors. Molecular docking. Molecular docking is one of the methods of structure-based drug design which predicts the accurate placement of the small lead molecule, ligand molecule within the active site of their target protein or receptor. Molecular docking is the most accurate method for the estimation of most favorable binding modes, for the estimation of the bioaffinities of the ligand with their receptor. Okay, so basically in this, the scoring function is done by the help of which the interaction between the ligand drug molecule or the lead molecule and the receptor can be observed, and this has been successfully used in the virtual screening techniques for the optimization of the lead compounds. Molecular docking process basically involve the three important functions to be evaluated, like it involve the virtual screening of the ligand molecule against the receptor. It involve the prediction of the binding pose, that in which direction the ligand can bind effectively with its target site, and we may check the strength of the affinity of the ligand and receptor. All these functions are evaluated by the molecular docking and can be expressed with the help of the different tools of the docking software, like search algorithm and the scoring function. These are for analyzing the conformation of the ligand molecule in the three-dimensional structure of the target receptor protein. Scoring function is basically the evaluation or the analysis of how well a drug binds to its target active sites. That is basically checked by the observing bioaffinity after predicting the binding pose of the ligand molecule with the receptor. So in this diagram you may see that this is the receptor and these are the different ligand molecule. In order to draw your attention, the ligand molecule are drawn in the different attractive geometric shape, like in shape of a star, circular, hexagonal and others. So basically what is done, these ligand and receptor, that is the protein, are now docked in the docking software or molecular docking software, so in which virtual screening is done. Okay, you may observe that screening of this molecule is done, and those which have the strong affinity with the receptor can bind effectively. Like in this case, these stars have the strong binding affinity with the receptor molecule, and this docking process is generally also based on the energy of these lead molecules, which have the lowest energy can bind effectively with the target receptor site. Now let's have a quiz for your self-assessment. You may post your answers in the comment section. Question number 1. Define computer-aided drug design. Question number 2. Name the two approaches or types of computer-aided drug design. Question number 3. Define structure-based drug design. So this is all about this lecture. Thank you so much everyone. Allah bless you all.